Welcome everyone to this chapter on the molecular basis of genetic disease, part 2. Welcome everyone to this chapter on the molecular basis of genetic disease, part 2. This presentation covers hemoglobinopathies, which are inherited disorders that affect hemoglobin in red blood cells. These conditions can impact the ability of hemoglobin to carry oxygen effectively or cause red blood cell damage. There are three main causes of hemoglobinopathies. The first one is structural alterations, and this is where changes in the protein sequence cause diseases such as sickle cell disease. There are thalassemias, where there is a decreased production of globin chains, and this leads to an imbalance and disease. And there are hereditary persistence of fetal hemoglobin, which is HbF for short, which is a benign condition where fetal hemoglobin continues to be produced right after birth. Changes in hemoglobin structure can have three effects. Hemolytic anemia, which is unstable hemoglobin, which leads to red blood cell breakdown. An example would be HB Hammersmith. Modified oxygen transport, this is where hemoglobin variants can affect oxygen affinity or form methemoglobin, which cannot carry oxygen, such as in HB Hyde-Park. And there are thalassemia-like variants where reduced production of globin chains result in disease in the patient. An example would be HBE disease. Sickle cell disease is caused by a mutation in the beta globin gene. This changes the shape of the red blood cells into sickles, and they can block blood flow. It's inherited in an autosomal recessive pattern, which means that patients will need two faulty copies of the gene to develop the disease. Sickle cell disease causes anemia, pain crisis, and organ damage due to blocked blood flow. Carriers of one faulty gene, known as sickle cell trait, are usually unaffected. However, they can face risks under extreme conditions, such as in high altitude. Mutated hemoglobin S polymerizes in low oxygen, causing red cells to become rigid and sickle-shaped. These cells block capillaries, reducing blood flow and causing organ damage. Sickle cells also rupture easily, releasing hemoglobin and worsening the condition. In the next few slides, I'll be talking about how modifier genes can affect disease severity in sickle cell. So high levels of fetal hemoglobin, which is HbF, can reduce the severity of sickle cell disease by preventing sickling. Studies have shown that variants in the gamma globin gene, BCL11A, and MYB account for differences in HbF levels, and they are therefore called modifier genes. BCL11A is a gene that silences the gamma globin expression after birth, reducing HbF levels. So by inhibiting BCL11A, HbF production can be increased, which can improve outcomes in sickle cell disease and beta thalassemia. And clinical trials have shown very promising results. The other modifier gene is MYB, and it's an important regulator of HbF. Interestingly, individuals with trisomy 13 show higher HbF levels due to two microRNAs, and they reduce the MYB expression. This decrease in MYB allows for greater HbF expression and illustrates how additional genetic factors such as microRNAs can modify disease severity in sickle cell disease. Unstable hemoglobins are typically caused by single nucleotide variants, and they lead to hemoglobin denaturation. The denatured hemoglobin forms Heinz bodies that damage the red cell membrane, resulting in hemolysis, which is the destruction of red blood cells. Certain hemoglobin variants impact oxygen transport without compromising hemoglobin stability. For example, HV Hyde Park leads to methemoglobinemia, while HB Kemsey locks hemoglobin in a high oxygen affinity state, affecting oxygen delivery to tissues. Methemoglobins occur when the iron in heme remain oxidized, as seen in HB Hyde Park, resulting in cyanosis. Meanwhile, high oxygen affinity variants like HB Kemsey limit oxygen release to tissues, often leading to polycythemia. Thalassemias are the most prevalent single gene disorders worldwide. These are disorders in hemoglobin synthesis that affect either the alpha or beta chains, resulting in an imbalance that leads to red blood cell destruction and anemia. There are two primary types of thalassemias. There's alpha thalassemia and beta thalassemia, and each impact a specific globin chain. Alpha thalassemia is particularly prevalent across Africa and Asia, while beta thalassemia is more common in the Mediterranean and Middle Eastern regions. In thalassemia, excess globin chains accumulate because of reduced production of one type of chain. These excess chains damage red blood cell membrane, causing anemia. Alpha thalassemia can be severe, such as seen in hydrops fetalis, where excess gamma globin forms ineffective hemoglobin. Milder forms cause gradual damage to red blood cells and anemia. Beta thalassemia, on the other hand, is often caused by single nucleotide changes. Severe cases require lifelong management, including blood transfusions, iron chelation therapy, and potentially bone marrow transplantation or gene therapy. When alpha thalassemia coexists with beta thalassemia, it can actually reduce clinical severity in the patient. This occurs because the reduced alpha globin production balances the globin chain ratio, preventing excess alpha chains from causing damage. Beta thalassemia results from DNA changes that reduce beta globin production. Some large deletions can disrupt multiple genes but lead to benign conditions, such as hereditary persistence of fetal hemoglobin. Some beta thalassemia variants produce mRNA that doesn't work because of premature stop signals. These mRNAs cannot be translated into functional beta globin. So common types of variants that cause stop signals are promoter and splicing variants, frameshift or nonsense variants. RNA splicing variants are the most common, disrupting normal splicing mechanisms. Some beta thalassemia variants produce mRNA that doesn't work because of the premature stop signal, as I mentioned before. These mRNAs cannot be translated into functional beta globin. When a premature stop codon is located far from the last exon, the mRNA is broken down by the cell. This reduces abnormal protein production but lowers the beta globin levels. mRNA needs proper capping and tailing to remain stable. So a variant in the beta plus thalassemia disrupts this process, reducing mRNA stability and beta globin production. So hemoglobin E is actually common in Southeast Asia. This particular disease reduces beta globin production and can cause severe anemia in the patient when inherited with another beta thalassemia allele. An example would be the HBeP dot glutamate to lysine, which is a common variant in Southeast Asia. This variant activates a cryptic splice site and reduces the beta chain synthesis in the gene. Compound heterozygotes, as I mentioned, with another beta thalassemia show severe anemia. Large deletions, on the other hand, in the beta globin cluster can disrupt normal expression but sometimes allow continued production of HbF. This helps compensate for beta globin deficiency. Thalassemia actually imposes a significant health burden in affected regions, and in those regions where it is prevalent, public health approaches such as carrier screen programs have been really important in reducing disease incidence. These initiatives have helped reduce birth rates of affected newborns by up to 90% through widespread education and genetic screening.